Good evening. So, just a short one today. It's going to be about self-fractionation. Now, this is one of the easiest things to do really well, but it's one of those things people actually do very badly on. In all honesty, it's just a load of facts we need to learn. Once we've learnt those facts, you will absolutely be able to get 5 or 6 marks on any 5 or 6 question about self-fractionation. So, we start with the definitions. You'll never be asked this per se. I just feel like you do need to understand it in order to be able to understand what we're about to talk about. So whatever we're doing here, we're trying to separate out the organelles within a cell. So the whole process we're about to talk about is about separating the organelles in a cell. So I'm going to want to do some research, and I'm only going to want to research mitochondria. If I only want to research mitochondria, then I have to only get the mitochondria out of the cell. And the way you do that is with self-fractionation, which removes everything else and leaves you with only mitochondria, or only nuclei, or only rough endoplasmic reticulum. So there's 3 words here that we're going to use. Self-fractionation, which is where we're going to break up our cells and get the organelles out. Homogenation, which is the actual process of breaking up the cells, usually in a blender, or we can often use a pestle and mortar. And ultracentrifugation, which is where we get a centrifuge and we spin the cells up so fast that they separate according to their mass. And we'll talk about that in a second. So self-fractionation is the name for what happens when you put together homogenation and ultracentrifugation. So first you homogenize your sample, then you ultracentrifuge it, and once you've done both of those 2 things, you have done self-fractionation. They go together. So let's talk about the process. Now, stage 1. First things first, you have to get your cells prepared properly. So you get some cells. Now, we don't need to know where we get our cells from. You could just get them off your cheek with a little swab if you wanted to. And you put them in a cold, buffered, isotonic solution. Now, let's talk about why that is. Cold, because it's going to reduce enzyme activity. Now, we only talk about temperature in biology if it's got anything to do with enzymes. So the reason why people die when they get a fever is because their enzymes denature in their brain. The reason why your body doesn't work particularly well if it gets very cold is because the enzymes don't work as well when it's cold. So with cold, we're just trying to reduce enzyme activity down. And there are plenty of enzymes within your cells that could damage organelles, could destroy organelles, and we don't want that. So you make them cold, and that stops the enzymes from working, and that stops them from damaging your organelles. Buffered. That means we're going to add a pH buffer, a chemical that keeps the pH the same, no matter what. Like if I've got pH buffer and I pour it in, the pH will stay at that pH. So I get my pH buffer 7, I put in my 7 buffer, and that will stay at pH 7. I can get a bottle of sulfuric acid, pour it in, and that buffer will make sure that the solution stays at pH 7. And I just pour in acid after acid after acid, stays at pH 7. That prevents protein damage. So the issue with changing the pH is, changes the pH break things like ionic bonds. If you break the ionic bonds in protein, it changes shape. So we don't want that. We want the proteins to stay the same shape because the whole point is to get these organelles out and be able to do research with them. And if their proteins are all broken, they're useless. So cold, stop the enzymes working, that would destroy my organelles. Buffered, stop my proteins from getting changed shape by changes in pH. And then isotonic. Now we should know the word isotonic from osmosis. Just means that there's the same amount of water concentration, the same water potential even, on both sides of the cell membrane. So basically I want a chemical that's going to have exactly the same water potential as the inside of my cell, so that osmosis doesn't happen. The water still goes backwards and forwards between the cell and the solution, but it goes at the same rate backwards and forwards, and so overall the amount of water inside the cell stays the same. So we're talking about isotonic solutions. Right, stage 2. Once we've put it in our cold to stop the enzyme, buffered to keep the pH the same so the proteins don't change shape, isotonic so that the cell doesn't gain, so that the organelles don't gain or lose water and burst or shrivel up. We go to stage 2, homogenization. We get our cells and the liquid, the cold isotonic buffered solution, and we blend them, usually in a blender, but sometimes in a pestle and mortar, to release the organelles. Okay, nice and straightforward. And incidentally, if you want to take any notes at any point, just pause me. I'm going to go through this quite fast. I'm going to do an exam question at the end. So, stage 3. Once you put them in a solution, cold buffered isotonic, and you've broken them up, then we do we filter them. Now, the point of the filtering is to remove insoluble material, things like cell walls, things like connective tissue, bits of ligaments and cartilage, things like the remains of the cell membrane, all the stuff we don't want. Okay, so we filter it to get rid of all the extra stuff, and we should be left with just the organelles once we've filtered it. And then finally, we get to the stage which is the most interesting. We put what's left inside an ultracentrifuge. So I should be left with a beaker or a test tube of pure organelles. All the nuclei, all the mitochondria, chloroplasts, whatever it might be, should be my beaker or my test tube. I'm then going to put that into a little test tube. I'm going to put that in my ultracentrifuge. I spin it. Now this is important actually. I don't actually really like this sentence. At a slow speed. No, it's still a very fast speed, still hundreds of rotations a minute, just at the slowest possible speed for an ultracentrifuge. Now that's still spun quite fast, but not tens of thousands of rotations a minute, just a few hundred rotations a minute. That's still more than one spin a second, which is fast, but not ridiculously fast. So the first thing that will happen if I spin it at the lowest possible setting is the nuclei will be forced to the bottom because they have the highest mass, and so gravity has the biggest effect on them. So as I'm spinning it, the centrifugal force happens, things get dragged towards the end of the test tube, heaviest things get dragged first. So the nuclei will be knocked out of the suspension, and you end up with a little solid pellet at the bottom. Those will be the nuclei. Now, you could just remove those if you wanted the nuclei, you could do studies on those, or you can take them out and put them in the bin. Then you can spin it faster to gain the next heaviest organelles, such as mitochondria and chloroplasts. And all you do is you spin it faster and faster and faster and faster until you're left with just the organelle that you want, So if I want ribosomes, I'm going to have to spin it loads of times because I'm going to have to remove everything else. If I want mitochondria, I'm only going to spin it twice because that's going to be the second thing that comes out. Now you're not expected to know what will come out first or second or third. The only one I'd expect you to know is that nuclei are the biggest ones, so they will come out first. Right, let's run through this one more time in terms of what people get wrong. People get cold buffered and isotonic wrong because they just fail to remember it. You need to make sure you're memorizing why I want a cold buffered isotonic solution. That's work you need to do. You need to sit down, look, cover, write, repeat, write yourself flashcards, whatever it takes to learn those facts, because you can't get marks in this question if you don't understand those facts. The second thing that goes wrong is people say that the cells are going to burst or shrink. I almost did that myself earlier. We care about the organelles, not the cells. We're about to shred the cells into a million pieces. So the cells can burst as far as I'm concerned. We're trying to prevent the organelles bursting when we're talking about an isotonic solution. Stage 2, people don't really forget that. That's good. You just break it up, you blend it. Most people remember that. A lot of people forget to filter. That can sometimes come with a mark scheme. Filtering is an important stage, otherwise you're just left with a load of bits in your solution. And then the fourth stage, ultracentrifugation. People write this, and I need to change this. I've stolen this PowerPoint of somebody else, but I need to change this because we don't spin it at a slow speed. We actually spin it at quite a high speed. Right, let's have a look at a question for this then. I'm going to skip through some stuff here. There you go. Describe and explain how cell fractionation and ultracentrifugation can be used to isolate mitochondria from a suspension of animal cells. So it's asking you, how can I get the mitochondria out of animal cells using cell fractionation and ultracentrifugation? 5 marks. Can you have a go from what we've just said, writing down the answer to that question? So pause me. It should take you about 5 minutes, but see how long it does take you. Pause me. Have a go. Right. Hopefully you did pause me. So here's the answers. There's 6 possible answers. 1, cell homogenization, i.e. blending the cells. 2, filtering. I told you people forget to write filtering. If you don't write filtering, you're throwing away marks. 3, use isotonic solution to prevent damage to mitochondria or organelles. Keep cold. Now that would include things like swelling and shriveling. You'd get the mark for that because that is damage. 4, keep cold to prevent reduced damage by enzymes. Use buffer to prevent protein enzyme denaturation. So they put both marks within one mark there, which is a bit harsh. But if you said you keep it cold or you use a buffer, then you will get the mark there. Centrifuge to separate nuclei at a lower speed, not a low speed, a lower speed. To separate nuclei, cell fragments, heavy organelles, and respin at a higher speed to get mitochondria. Now, why I've read that to you, I'm not sure, because I think that's a very clear mark scheme. It's just one of those ones you should be correcting your answer, adding anything you didn't write, and then memorizing this. It's the first thing I would do is I would memorize this because it's really important. And in terms of how you're going to memorize this, write it on a piece of paper, check if you were right, put it to one side, write it again. Just write it over and over and over again until you can't get it wrong. Do it over days and days and days. And do this for every exam question that you think is going to come up. This one is very likely to come up at some point. Right, that's me. I think that's quite a nice, quite short one as well. So hopefully that's helped you to understand self-fractionation.